Okay, firstly, I will talk about the origin of mitochondria and chloroplasts. Mitochondria and chloroplasts likely evolved from engulfed prokaryote that once lived as independent organisms. At some point, a eukaryotic cell engulfed an aerobic prokaryote, which then formed an endosymbiotic relationship with the host eukaryote. And then gradually developing into a mitochondrion. Eukaryotic cell containing mitochondria that engulf photosynthetic prokaryote, which evolved to become specialized chloroplast organelle. So that's how mitochondria and chloroplasts form. Next is I will talk about the function and structure of chloroplasts. As you can see, chloroplasts are the site of photosynthesis in eukaryotic cell. They are only present in photosynthetic cells like plant cell and algae. The structure of chloroplast can be found. The structure is chloroplasts can be found in the cell of the mesophyll in plant leaf. They are vesicular and have a colorless center. The size of the chloroplast also varies from species to species, and it is constant for a given type cell. Okay, the function of chloroplast is to absorb of light energy and conversion of it into biological energy. The most important function of chloroplast is to make food by the process of photosynthesis. Production of ATP by photophosphorylation. Next is light reaction that take place on the membrane of the thylakoids. Also dark reaction, or known as Calvin cycle that take place in the stroma of chloroplast. Next is I will talk about the structure in chloroplast. This is inner membrane, thylakoid, granum, outer membrane, stroma, lamellae, stroma, fluid filled space, and chloroplast ribosome. Okay, the outer membrane is semi-porous membrane and is permeable to small molecule and ion, which diffuses easily. The outer membrane is not permeable to large protein. The inner membrane of the chloroplast form a border to the stroma. It regulates passage of material in and out of the chloroplast. Stroma is an alkaline aqueous fluid which is protein-rich and is present within the inner membrane of the chloroplast, while stroma lamellae is the region of the stroma that interconnects the thylakoid stalk that are called as stroma lamellae. Okay, next is thylakoid system. The thylakoid system is suspended in the stroma. The thylakoid system is a collection of membranous sacs called thylakoids. The chlorophyll is found in the thylakoid and is the site for the process of light reaction of photosynthesis to happen. The thylakoids are arranged in stacked down called as grana. So each granum contains around 10 to 20 thylakoids. So next is the genes that play important roles in the function of chloroplast and the function of the gene. The genes are RBCL, MATK, NDHG, and ITS1. Each of the genes function differently. So RBCL is for photosynthesis, MATK to encode protein, and NDHG is to generate energy, while ITS1 is non-coding spacer. Okay, there are two commonly used genes and two rare genes, which is RBCL and MATK are commonly used genes, while NDHG and ITS1 are two rare genes. So the function of each gene is for RBCL to code for first major step for carbon fixation, while MATK is to encode protein in an organelle. While NDHG is to generate energy, NDH shuttle electron from NADPH via FMN and iron sulfur center to quinones in the photosynthetic chain and possibly in a chloroplast respiratory chain. The immediate electron receptor for the enzyme in this species is believed to be plastoquinone. Couple the redox reaction to proton translocation and thus conserve the redox energy in a proton gradient. Okay, for ITS1 is 2 non-coding spacer. Okay, next is RBCL, MATK and NDHG are protein coding gene, while ITS1 is non-protein coding gene. Okay, there are 2 genes that are more commonly used. Why it's commonly used than the other 2 genes? It is because RBCL RBCL is a part of DNA sequence that located in cpDNA or chloroplast DNA and has a chance to be used as a DNA barcode because this coding region is given universality and is in amplifying and analyzing. This gene provides many characters to study phylogenetic because it has full length approximately 1400 BP. So that's why it is commonly used in lab. That's all from me. Thank you.